Hi, today we're talking about CRISPR, and it's a biotechnology used for genome editing. So what is CRISPR?
So CRISPR, or CRISPR-Cas9, stands for Clustered Regulatory Interspace Short Palindromic Repeats. But because no one likes lifting off that mouthful every time, the acronym CRISPR is used. So CRISPR acts like an enzyme that cuts genomes like a pair of scissors and allows a new slice of DNA to be exchanged. So now that we know what it is, how does it work?
Well, this is how it works. So there are target bacterial cells that contain large sections of repeating DNA sequences. So in between those sequences, there are these things that are called spacers, and those spacers match virus DNA sequences. So when it's exposed to viral infection, the DNA is transcribed into RNA and it instructs the nucleus to cut the viral DNA. So CRISPR has worked hard for several years on the way to control the cutting to edit the genome. So what are the positive things about this?
There's a lot of positive things about CRISPR because it can be a possible treatment for genetic diseases that we don't have any treatments for, such as cancer and diabetes. Those are monogenic and polygenic diseases, so CRISPR can actually cut that gene out. It's very precise. It's more precise than any other genome editing techniques that we've found so far because it can target a single cell. And the technology is pretty simple to use and it's very scalable. So if we wanted to use it on a wide scale, we could do that pretty fast. So why wouldn't we be using CRISPR?
Even though CRISPR is a very powerful tool, it also comes with disadvantages and limitations. One is that it requires a bulky and big protein. This can be recognized as foreign in the immune system. It's also not 100% accurate or efficient. This can have consequences later on. It is also difficult to deliver in mature cells. This can become an issue in clinical applications. And lastly, there are ethical dilemmas that can result in bias in society.
Ethical dilemmas related to the use of CRISPR in humans arise when the genome of germ cells and embryos are edited. Altercations in the genome of reproductive cells can pass on and potentially alter the future genome of the human species. As far as the use of CRISPR on embryos, traits that are deemed unappealing may be selected against and altered before conception. Will CRISPR or similar technologies be used to advance and normalize a new form of eugenics? If this happens, this could lead to further institutionalized racism, sexism, and even classism. Here is a brief list of our references, followed by a short animation of some of the risks involving CRISPR.